Okay, team. Today we will finish up the unit by discussing DNA replication and protein synthesis. You can see where we're at in Module 5. There's the genetics PowerPoint. We've already finished that. You should be working on these sheets, as I mentioned. And here is what will be the last video of this unit. Okay, so we start out, we talked about, well, the DNA is replicated, and we said that occurred in the synthesis phase of the cell cycle, but we kind of left it at that. We didn't really go into any sort of detail about how that happens. I mean, if you think about it, there are billions of base pairs in the DNA molecule in a human being, and each of those billions of base pairs in the sequence that they occur is replicated. And so, we want to understand, how does that happen? That would be our goal. Okay. So in order to understand this, let's think about the structure of the DNA molecule, first of all. And we can see here that the structure of the DNA, if you recall, it's this helical shape, but it, see if I can move this. You can't see it, but I can, and it's driving me crazy. I'll move it up here for now. The DNA molecule consists of these sugar phosphate bonds, right? And then what holds them together are the nitrogenous bases. And so you can see here, for example, is the sugar, right? And what's the sugar is deoxyribose, right? There's a sugar, there's the phosphate group right there, sugar, phosphate, sugar, phosphate. Same thing down the other side. And again, we're saying these kind of form the sides of the ladder. And then what holds them together, as you recall, are the nitrogenous bases. And you can see these are represented here. So there's a nitrogenous base and there's one. And maybe if that's an A, then that would be a T. And if that's a T, then that would be an A, etc. So remember, A's and T's and C's and G's go together. And these are hydrogen bonds here that hold the two sides of the helical molecule together. Okay, so that's just kind of a refresher in order for you to understand sort of how this works. And so what happens when the DNA replicates, there are some enzymes involved, first of all. And I think it's worth it to, at least be aware of these enzymes. And one of the enzymes is called helicase. And remember, the DNA is helical in shape. What this helicase enzyme does is it literally unravels that spiral that is the DNA and makes it linear, the way we've been drawing it. So that's important. And then what you can see happens quite literally, and I'm going to draw my own little piece of DNA over here. I don't really have a lot of room, but I might have to go to a blank screen, actually, in order to show you what happens. But we'll come back to this slide. I think that makes the most sense. But while we're on this slide, the other enzyme that's important is called DNA polymerase 3. So both of these enzymes are important. The DNA polymerase 3 is what guides this, what they're calling the complementary number 1 and complementary number 2 into their appropriate positions. And so quite literally what happens is you could think of the DNA molecule like a giant zipper. And when the replication process occurs, it literally unzips, put a little fly on there, and it literally unzips from one end to the other. And in the process of doing that, each side is used as a template to make the other side. And so I'm going to show you how that works. But this image is nice because it shows you the names of the enzymes there. Okay. Okay, let's just go to this blank screen for just a second, and that will allow me to show you in a little more detail what's going on. So let's start by making a little piece of DNA, and just be so that we can be clear about how this works and we know how the molecule is set up. If I give you this side, like this, you could give me the other side, I would hope, at this point. Okay, so if you got an A on this side, you're going to get a T. If you get a T, you're going to get an A. A's and T's, C's and G's. If that's a G, you're going to get a C. T, T, G, C, A, T. Okay, so now we've made a little piece of DNA. Again, there's a billion base pairs in human DNA, billions of base pairs. This is just a little fragment of our DNA, for example, and now you can see here's the hydrogen bonds holding these all together. Okay. And like I said, it literally unzips from one end to the other. So the molecule, if you looked at the next picture, it would look like this. It comes apart like this. And in the process of coming apart, let me just write these in. Make sure we don't have any mutations. So there's one side, and here's the other. Okay. And then what happens is a complementary strand will slip in on this side, like this. And a complementary strand will slip in on this side, like this. And what they'll do is they'll use either side as a template to make the other side. And so, you know, if there's an A there, it's going to be a T. And if there's a T, it's going to be an A and a C and a G and a T and a T and a C and a G and a G and a T and an A and a T. Okay, so there's the one side. The same thing happens on the other side. So what these red lines represent are just the sugar phosphate bonds of this sort of blank strand of nucleotide. Okay, and then same thing on this side, but we're using the other side as the template. So if there's a T, you're going to get an A. And here, if there's an A, you're going to get a T. And look what happens. We end up making two replicas, right? So now look what we've done. They refer to this process as being semi-conservative. Can you think of a reason why they would say it's semi-conservative? What's being conserved in this process? What you'll note is that we still have both sides of the original DNA, don't we? So now the two replicas consist of an old side, the original side, we'll call it, and the new side, and then the same thing here. Okay, so it's semi-conservative in that it conserves the original, right? And then ultimately you end up with two identical pieces of DNA. And I'm not going to take the time to describe those, but you understand. So this is the replication process, okay? And again, when does this occur? It occurs in the synthesis phase of the cell cycle. That's where this process occurs. it's a very accurate process, but you could imagine there could be a little glitch, right? And if there's a little glitch here, let's say something happens to this location, this base, well, that's called a point mutation, and we're going to talk more about what happens in point mutations and why they can be very detrimental to the organism. Okay. So we'll stop there with that. And if we go to this slide, we're going to now talk about protein synthesis, okay? And protein synthesis is really, honestly, it's really fascinating. Sorry, oh, darn, okay, well, sorry. Things are all haywire now for me. Okay, back to this slide. So protein synthesis involves making a protein, and the primary structure of a protein anyway, out of amino acids, right? And here's the thing, the DNA, believe it or not, is a recipe for making proteins. And the way that it's a recipe is that the sequence of bases in the DNA, oops, determines the sequence of what in a protein? Of amino acids. Okay, what I'm saying is the base pairs, the bases determine the sequence of amino acids. They're like a recipe for making a protein. Let's see how that works. Now, it's similar to replication in that the complementary sides of the DNA come apart, but rather than coming apart from one end to the other, they come apart at some location within the length of the DNA molecule. So they're still attached on this end, and they're still attached on this end. It's just it opens up at some location. That location is dealing with a gene or genes, okay? That information is going to be copied, okay? And the way that the way that it's copied, what we call transcription, is done by the other nucleic acid that we haven't really talked about, which is RNA. So RNA is going to come into the picture. And here's the thing about RNA that you might as well just get in your head. No thymine on RNA. No T's on RNA. Instead, you get the fifth nitrogenous base called uracil. This is very important, okay? RNA, it turns out, is a single-sided molecule that does not contain thymine. In the place of thymine, it contains uracil. So now if you look at what's happening, this thing is popped up now. I don't know what it wants from me. There we go. If you look at what's happening here, the DNA is opened up. Here's a messenger, what we call messenger RNA, and it's slipping in and copying this information. We call it transcription. To transcribe means to copy. But you'll notice where there's an A, here, you're not getting a T, now you're getting a U. And we're going to go back to the whiteboard, and I'm going to show you a good example of this in just a second. Okay. Now here's the thing: the DNA has two sides, right? And depending upon which side the messenger RNA slips in and copies or uses as a template, you're going to get two different sequences. You're either going to get the sequence or the complement of the sequence, right? And so what we like to say is that there's a reading side to the DNA and a non-reading side. And I don't know how the messenger RNA knows which side is which, but safe to say that it knows which side to read and which side not to read, because it's only using one side or the other. Okay, so if you see this in your reading, something about reading and non-reading sides of the molecule, you'll understand what's going on. Okay, so let's move on from there, and let's say that we're going to make a little piece of a protein out of a little piece of DNA. Okay, that's the goal. So here's the thing. We're going to make a little piece of DNA. And I just have to look at this for a second to make sure that I use the right sequence. AUG, so it would be TAC. Okay. Okay. Don't worry, it's all going to make sense in a second, I promise. Okay, so here's a little piece of a DNA molecule. We're going to start with TAC. Oops, shoot. Look what I've done now. Okay. Making a little bit of DNA. Let me just make this CGA. So, okay, sorry about that. Has to work out or it doesn't work out. Okay, good. So now here is one side, we could say, of the DNA molecule. And we're going to let this be the reading side. Okay. This is the important side of the molecule. You could give me the other side, easy enough. If there's a T, you're going to get an A. If there's an A, a T, a C, a G, a C. You could do that already. We're going to say this is the reading side of the DNA molecule. What is going to then happen is, if this isn't the information for making a protein, then the DNA will open up at this location and in comes a strand of messenger RNA. So we're going to show the strand of messenger RNA slipping in. We're going to label this messenger RNA. Okay. And now we're going to transcribe. We're going to do transcription. We're going to use this to make the messenger RNA. Now remember, if you have a T on the DNA, you can get an A, right? But if you have an A, you can't get a T on RNA. You're going to get a U instead for uracil. We have a C, we can get a G. A G gives us a C. So those don't change. But what about here? U, U, A, right? G, G, C, U, 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 C, C, U, U, G, A. Okay? Now we have made the messenger RNA. So what then happens is, where are the proteins made? And why do we call it messenger RNA? Well, the recipe book is in the nucleus, but the proteins are made. What is that organelle where the proteins are made? Called the ribosome, right? So now this messenger RNA, it slips in, it copies the recipe, it slips out, and it's going to the ribosome. It's carrying that sequence of information to the ribosome, okay? Each 3-base pair on the messenger RNA is called a codon. So we got 1, 2, 3, 4, 5, 6, 7 codons on our little strand that we made. And each codon represents an amino acid. This is really important, okay? Each of these 3-base sequences on the messenger RNA represents a specific amino acid, okay? And we call this the genetic code, and the code has been broken. That's the cool thing about it, is that we've broken the code, okay? So let's look. We have, for our first codon, AUG, and there is a genetic code right here that tells us, if we look up AUG, and the way this works, I know you can't see the pointer. Let me make it to where you can see the pointer for a second, okay? So the way this works is, this is the first letter of the 3-base sequence. This is the second letter, and this is the third letter, okay? So our sequence was AUG, so here's the first letter A, that means it's gonna be somewhere in here. Second letter U narrows it down to this area, and then G comes across here, and you can see this particular amino acid is called methionine, or MET, it's abbreviated MET. Okay, now here's the thing, there are 20 different amino acids, right? But there are 64 codons. So what that means is there's some repetition. For example, we have many more codons than we have amino acids. And so there are 64 different possible combinations of those 3 letters. So you can see what that means is there's repetition. For example, all 4 of these codons bring arginine, as do these 2. So there's actually 6 different codons that represent the same amino acid. Okay. Now here's the thing, MET is very important. Methionine is important because it's always what we call the start amino acid. All proteins start with methionine, and it ends up they get sometimes cut off later. Okay. So our first codon, AUG, represents methionine, and so we can go back to our sequence, and we can write MET right here. This is the protein that we're making. Okay, so there's MET. Now the next codon we go to is CGC. Let's write a couple of these down, CGC and then UUA, and we'll look up both of those. CGC and UUA. CGC and UUA are the next 2. CGC, CGC is right here, and that represents arginine, ARG. And then UUA, where's UUA? We got a U and a U, and an A is leucine, LEU. Okay, so let's write those 2 down. Arginine and leucine are the next 2. Oops, shoot. Did not mean to do that. Get that back. Okay, so now we have met. We have arginine, ARG. UUA was leucine. See how it works? We're building this sequence of amino acids in the protein by using these codons of the messenger RNA and looking in the genetic code. You don't have to worry about trying to memorize the genetic code. Good Lord, I'll give you the genetic code if you need it to look up the amino acids. I did jokingly say, you might want to go ahead and memorize the genetic code. That was a joke. One poor student tried to memorize the genetic code. She was very upset when the test came and I gave her a copy of the genetic code. So don't be that person. If I try not to say stuff off the cuff anymore because I realize people take it literally. Okay, let's keep going. Now we have GGC and UUU. Okay, let's go back to our other page, GGC and UUU. Let me just write those down so I remember. GGC and UUU. Let's see, GGC, there's G. GC is glycine right here, GLY. And UUU should be easy to find. It's phenylalanine or PHE. So glycine, phenylalanine. GLY and PHE, and we're almost done now. We got CCU and we got UGA. So we got two more, CCU and UGA. CCU, let's see, there's CCU is proline, PRO. And then look, UGA is right here. And what does that say? Stop. Stop is not an amino acid. Stop is the signal to the ribosome that, hey, we're done. We're done making our protein. This is the end. It's time to spit the protein out. So all proteins end up starting with the amino acid met or methionine, but they all also end with stop, okay? And oftentimes what will happen is the information will be copied. The information will be actually copied. Multiple proteins will be copied on one messenger RNA. So what then happens is when the ribosome gets to stop, it knows, okay, that one's done. And then if it gets met as its next amino acid, it knows we're starting a new protein. So that's very convenient. And now I can't remember what it was. I think it was proline. Must have been proline and stop. I'm going to go with that. Okay. So now we've built our amino acid. PRO, and then stop. Okay, so look what we've done. We used the messenger RNA codons to determine the sequence of amino acids in the protein. And where did that messenger RNA sequence came from? Well, it came originally from the DNA through transcription. Now, there's one last piece of the puzzle that I got to tell you about. And that last piece of the puzzle is, how did these amino acids get where they're going? Right? How do they get there in the ribosome? The ribosome comes and reads the first codon. How does MET magically arrive? Well, that's what I'm going to explain. There's another kind of RNA called transfer RNA. And what transfer RNA does is, as the name implies, transfers the appropriate amino acid to location. Okay, how does that work? Well, the way that it works, I'm going to do a different color. Let's do blue just for fun. This is the tRNA. Okay, and if this is the codon, this is called the anticodon. And so this is the complement. And these molecules are very short, very small. They're almost shaped like a keyhole like this. And what they have is they have a bunch of bases like this, and then they have an attachment at the bottom for the appropriate amino acid. And the other part that's important is right here at the top. This spot here is called the anticodon. In this case, it would be UAC. The anticodon is the complement of the codon. Check it out. What's the complement of this? AUG. A gives you a U. U gives you an A. G gives you a C. So there's the anticodon. And look what it's carrying. It's carrying MET. This ensures that only the appropriate amino acid ends up at the appropriate spot, because there's only one anticodon for every codon, right? Think about it. The codon is the complement of the DNA, right? The anticodon is the complement of the messenger RNA. So it's also the complement of the DNA. So really you can kind of cheat and look up here and see it's just the same sequence as this, except where there were T's, there's going to be a U now. So what literally happens is messenger RNA reads this codon, it signals this anticodon comes in and literally binds to it for a second, and it's carrying MET, okay? And there it sits. And then the ribosome goes and reads the next codon, and it says, okay, the anticodon here would be GCG, right? And that particular anticodon transfer RNA is carrying arginine. And now this is very important and very cool. At this point, we have two here. We have two amino acids beside each other, and this is where a chemical bond forms right here. And this particular chemical bond is called a peptide bond. And the peptide bond is the bond that holds amino acids together. At that point, y'all, at that point in the game, this transfer RNA lets go of MET and it leaves. Okay. And then we continue down the line. Next we have UUA, so that would be AAU. It's carrying leucine, peptide bond forms, and then this transfer RNA leaves. Where is it going? It's going to go pick up another arginine, right? And come back to do its job. Same thing happens here. We have, oops, shoot. We have CCG. It's carrying glycine, another chemical bond. We have AAA. It's carrying phenylalanine, another peptide bond. GGA, carrying proline, and then stop. Okay. This explains how in the world these amino acids get where they're going. And the answer is the transfer RNA. So here's the thing. If I give you the DNA and I give you the genetic code, I give you the reading or the non-reading side, it doesn't matter. I give you either side, you could make the other side, right? But let's just say I give you the reading side of the DNA, okay? You could give me the messenger RNA codon. You could give me the transfer RNA anticodon. And then you could give me the amino acid sequence. Now, here's the tricky thing. Here's where students make mistakes. The genetic code is based on the messenger RNA codon, okay? Not the transfer RNA anticodon. So let's just say you forgot that, and instead of looking up UUA, which would bring leucine, you look up AAU instead. You're looking up the anticodon instead of the codon in the chart. What's going to happen? Instead of AAU, you look up UUA, and you get leucine. You get a different amino acid. Okay, so you have to be really careful. Oh, I had it switched, sorry, but you get the idea. Don't look up the anticodon in the chart. You may get the wrong amino acid. Look up the codon. The other mistake you're likely to make is putting T's on any RNA molecule. There are no T's on RNA. U's instead of T's. There can be A's, but no T's, okay? Those are the two big mistakes that you might make, okay? So that's how proteins are made, right? They're made in the ribosomes. The recipe's in the DNA. The recipe is transcribed by messenger RNA, right? The ribosome then translates that recipe into making a protein using transfer RNA to bring the appropriate amino acids to their appropriate location. All proteins will start with met, and they'll all end with stop, okay? Cool. Now, what we might do, there's tons of animations, you know, for this kind of stuff. And what I'm trying to do is get this thing out of the way that you can't even probably see. That's in my way. Let's look at this. There's some really cool ones. I really like this one. It's 2 minutes and 25 seconds long, and I always show it to my class. So let's just take a second to watch. Oh gosh, we have to watch a Tom Tillis ad. Sorry.
Prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit, formylmethionine tRNA, and messenger RNA. The 50S ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA-carrying amino acids can bind. One is called the peptidyl, or P site, and the other is called the acceptor, or A site. There is also a third site called the exit, or E site, where transfer RNAs are released. The initiating transfer RNA carrying formylmethionine binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The formylmethionine carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site by a peptide bond. The ribosome now advances a distance of 1 codon, and the transfer RNA that carried the formylmethionine is released at the E site. A transfer RNA carrying the next amino acid now moves into the A site, where the anticodon on the transfer RNA matches the codon on the messenger RNA. The ribosome shifts down by a distance of 1 codon. As the shift occurs, the 2 amino acids on the transfer RNA in the P site are transferred to the new amino acid, and the second transfer RNA is released from the E site. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when a stop codon moves into the A site. A stop codon does not specify an amino acid and does not have a corresponding transfer RNA. The ribosome dissociates into the 30S and 50S subunits, and the messenger RNA and protein are released.
Introducing incognito. Tastes like meat made from plants.
There you go. That wasn't so bad, was it? Now you know how proteins are made. And so at this point, we are at the end of our discussion for this unit, which is the unit material for test 3. Okay. So the only other video you may or may not have seen is a video which is me working on one of these genetics problems sheets. And I'm going to show you which sheet I'm going to work on in case you want to work on that sheet as well. That's not it. I think it's the one that says still even more genetics problems. Yeah, so this is the one I'm going to actually do. I'm going to actually answer in a video. The other two you can just work on. You should, the questions are the same. The wording is pretty much the same. The organism might be different, whatever. At any rate, work this sheet before you watch the video. I think that makes the most sense. And then, you know, we'll be rapidly approaching the next unit test. Thank you. I will see y'all next time. I hope you have a great day. Bye-bye.